Hello everyone, today we will study about totipotency. Totipotency is the ability of a single cell to divide and produce all of the differentiated cells in an organism. Totipotency is the genetic potential of a plant cell to produce the entire plant. Isolated cells from differentiated tissue are generally non-dividing and quiescent. To express totipotency, they undergo dedifferentiation and then redifferentiation. In Latin, totus meaning entirely and potens meaning having power. History of totipotency. Haberland gave the term totipotency in 1902. Haberland hypothesized that entire plants could be generated from somatic cells. He believed that each cells come from a fertilized egg and is generally encoded, so it is capable of generating the entire plant body. Then, in 1950s, Stewart proved cellular totipotency. Cellular totipotency is a property of plant cells because a differentiated plant cell retains its capacity to give rise to a whole plant. In animal cells, however, the capacity for regeneration is lost after differentiation. Stewart and his co-workers in 1950 succeeded in growing carrot plants from isolated phloem cells. Here they take a carrot and then cut its transverse section of carrot root. Then they cut it into small fragments. Then fragments were cultured in nutrient medium. Stirring causes single cells to shear off into the liquid. Then these single cells in free suspension begin to divide. Embryonic plant developed from a cultured single cell. Plantlet was cultured on agar medium. Later it was planted in soil. A single somatic carrot cell developed into a mature carrot cell. Then Borgin and Nest confirmed totipotency of pollen grain in 1967. Then Nagata and Takibe in 1971 demonstrated totipotency of isolated protoplast. Differentiation of totipotent plant cells. Totipotent cells undergo differentiation and produces embryogenesis, organogenesis, histogenesis or cytodifferentiation. In embryogenesis, they produces embryoid and embryoid give rise to complete plantlet. In organogenesis, they undergoes callogenesis, rhizogenesis and callorhizogenesis. In callogenesis, only shoots are developed. In rhizogenesis, only roots are developed. In callorhizogenesis, shoots with roots are formed. Histogenesis or cytodifferentiation. In this, xylem, phloem are developed. High ratio of cytokinin to auxin leads to shoot development, that is coilogenesis. Low ratio of cytokinin to auxin leads to root development, that is rhizogenesis. Culture systems. Callus culture. Callus is an unspecialized, unorganized growing and dividing mass of cells. Can be grown indefinitely. It is suitable for cytodifferentiation, no photosynthesis and grows in dark. Can be used to isolate single totipotent cells. Many cultures lose their potential for differentiation during continual subculture due to epigenetic changes. Steps involved in callus culture. Firstly, we take an explant and these explants are washed with 70% alcohol. Then they are treated with sodium hypochlorite, then with mercuric chloride. Then the explant are cut into pieces. Then they are inoculated and callus are formed. And then callus are cultured in suspension cultures and new plants are formed in suspension cultures. Suspension cultures. Type of suspension culture in which single cells or small aggregates of cells multiply while suspended in agitated liquid medium. Possible to analyze biochemical pathways related to differentiation of cells. Single cell culture. Method of growing isolated single cell aseptically on a nutrient medium under controlled conditions. Can be isolated from a variety of tissues and organ of green plant as well as from callus tissue and cell suspension either mechanically or enzymatically. Single cell culture is an ideal system for study of biotransformation. Protoplast culture. Protoplasts are naked plant cells without the cell wall, but they possess plasma membrane and all other cellular components. The protoplast in culture can be regenerated into whole plant. It is easy to perform single cell cloning with protoplast. Let's have a look in this picture. Firstly, we take a plant and then we take explant. In this case, we take leaf as explant. Then we sterilize the leaf. Then we do epidermis peeling. Then peeled leaf segments. Then plasmolyzed cells in an enzyme mixture. Then partial cell wall degradation. Then pellet of protoplast. Then we centrifuge it. Then pellet of protoplast we get. Isolated protoplasts are treated with N2NO3 treatment. Then they undergo fusion. Fused protoplasts are formed. Then wall formation occurs. Then they undergo first division. Then clump of cells are formed. Then colony is formed. Then callus tissue is formed. And then callus undergo differentiation. And in this way, plantlets are regenerated, and regenerated plants are transferred from test tube to soil. And this process is repeated in many times. Then some factors that affect totipotency. Source of explant. Organ that served as tissue, source, physiological and ontogenic age of organ, season in which explant is obtained, size, overall quality of explant, nutrient media and constituents. Inorganic, macro and micronutrients, carbon, energy source, reduced nitrogen source, plant growth regulator. Culture environment, physical form of medium, presence absence of agar, pH of medium, light quality and quantity, temperature, relative humidity, gaseous atmosphere in the vessels. Importance, vegetative propagation, genetic modification, seed dormancy, embryo rescue, haploidy, protoplast fusion, germplasm preservation. Significance of totipotency. In plant cells, cell and tissue culture is reconstruction of plants from totipotent cells. It provides lot of information during differentiation in culture. The potentiality of somatic cells has been exploited in vegetative propagation of many economical, medicinal as well as agricultural important plant species. It helps in production of homozygous diploid plants through haploid cell culture and somatic hybridization and mutation. It can be utilized successfully for germplasm conservation and endangered plant species. Thanks and happy learning.